Our speaker today, I'm very happy to introduce Dr. Stephan Peischl. Dr. Peischl did his PhD in University of Vienna. He's majored in mathematics at that time. And then after a short postdoc also in Vienna, he moved on to the University of Texas at Austin and worked together with Mark Kirkpatrick. And after that, he moved to University of Bern, started as a postdoc, and since 2015, he's promoted to a research assistant, and he's still in this position at the moment. Stephan is a theoretician. He has broad interest in evolutionary biology and population genetics, and his main area of interest are the interactions of ecology and population genetics, the theory of speciation, and adaptation to spatially and temporally changing environment. Okay, Stephan, please start. We are looking forward to your talk.
Okay. So first, thanks for the invitation. This looks like a really exciting seminar series that somehow I have missed so far. So I'm looking forward to attend many of these meetings. Okay, and so today I will talk about a couple of projects, or like one larger project that actually started during my postdoc with Mark Kirkpatrick. So I'll, at the end, if there's time, come back to this. But broadly, I will talk about evolutionary rescue and the role of migration or gene flow on evolutionary rescue. And if anything is unclear, so I don't know the background of the audience, if anything is unclear, please interrupt me, write it in the chat, or just ask the question at any time. Okay, so before I start, I want to already acknowledge Mateo Tomasini, who was a PhD student with me at the University of Bern and is now doing a postdoc in beautiful Sweden, working on species range evolution. And this is a large part of this is based on these three papers. So lots of work was done by Mateo. So let me just briefly start with what is evolutionary rescue. So evolutionary rescue has gained a lot of attention in the past decade or so. It was started, the idea has been around for a long time, but in evolutionary biology, it was first explicitly modeled in the 90s by Richard Golmkiewicz and Bob Holt in a now seminal paper. And the idea is simple. So you have, so in this figure here, so the red line shows the population size, the abundance, let's say abundance of bacteria or just whatever you're studying. And then at some point, an environmental stress kicks in. So and this leads the population size to decline. So think of, this can be many different things. So this has been studied under different names in medicine, like the evolution of treatment resistance. So you apply antibiotics and the number of bacteria go down. In agriculture, you would call this like the evolution of pesticide resistance. And also in conservation biology, when we think about climate change, so the climate is changing and the population size declines. And then we have this blue line here that shows the allele frequency of an adaptive allele. So this is an adaptation to the new environmental conditions. And if such an mutation can spread quickly enough before the population goes extinct, then we will have this typical U-shaped trajectory in population size. So we have to decline and then an evolutionary rescue event and then an increase again. And I would argue that this is one of probably the theory of evolutionary rescue in single well mixed populations is very well understood. And most of the theoretical predictions have been confirmed mostly in laboratory settings. So there's a very nice review by Graham Bell where lots of this research is presented. And so recently this is gaining a lot of focus because of the evolution of antibiotic resistance, which might be future or is already like something that's challenging us in hospitals and also in the food production. And here's a chart from the bug from the Swiss Institute for Health, and it just shows the amount of antibiotic resistance that we find in clinical settings, and we see this increase in many of these organisms. We see that there's an increasing amount of the observed antibiotic resistance in clinical settings. So this is definitely something that we should like keep in mind. And since I'm now talking about antibiotic resistance, so part of the motivation for this work also comes from the study of the evolution of treatment resistance in antibiotics. So, and Pseudomonas, like a very important bacterium that causes lots of trouble in hospitals because it has a very high ability to develop resistance to antibiotics. And one interesting observation is that these bacteria, if they form biofilms, then the chance that they develop some form of resistance to the antibiotic treatment, so this would just be an evolutionary rescue event from the perspective of the bacteria, increases by several orders of magnitude. So this is a massive problem, and people really try to prevent the formation of biofilms in patients infected with Pseudomonas, because then antibiotics have you have a much higher chance for antibiotic resistance to develop. And the interesting part is that so biofilms have a very complex and intricate spatial structure. So we can see this here in this image, and here's a cartoon showing how these bacteria built this mucus layer around them and have this intricate spatially structured populations. And this has two effects. First of all, there's some kind of spatial structure, so we have a structured population now, but also this mucus is like a boundary to antibiotics. So we have the antibiotics have to diffuse through this mucus layer. So we suddenly have a gradual change in environmental conditions, and we have this spatial structure with movement between different locations. And importantly, I also want to highlight this paper that really shows that it's not purely this boundary effect of this mucus layer that leads to resistance. It facilitates the evolution of resistance, but also there's really a genetic adaptation underneath this. And on the experimental and modeling side, so there's a very interesting paper from Graham Bell and Andy Gonzalez from 2011, where they started a meta-population of yeast that they exposed to a salinity gradient. So this is this image here. So it should mimic like the species range where you have gradually changing environmental conditions. And then they shifted these conditions, so increasing salinity more and more in each of these wells. And they allowed for migration between these wells. And then we can see here in this plot, so we have different rates of environmental change. And on the y-axis, we have the number of rescue events observed. And the darkest one is no migration, no gene flow between these wells. And the other ones are two forms. So the gray one and the light one are local and global migration. So this means either migration just between neighboring wells or migration. So you take a sample from all the wells and then distribute it among all the wells. So essentially there's no real spatial structure, but population structure. And so there's an advantage, there seems to be a very clear advantage of migration to adapt to these changing environmental conditions, which can be seen as a little bit counterintuitive, because usually migration should hamper any form of local adaptation. At least that's one common perception. And Hildegard Uecker and colleagues in 2014 studied this system in a mathematical model. So they had this so-called island model. This corresponds to the global migration, where they could really show there is, so this is the probability of rescue versus the migration rate. And they found this intermediate range of migration rates where rescue is most likely. And the way they studied this was using simulations, and they also found some analytical predictions using time-dependent branching processes that can be applied in some scenarios. So it's a very detailed paper with lots of nice results. And we wanted to build on this, but look at a simpler model, because what we, or what Mateo and I wanted to figure out, we wanted to find under which conditions, because this is not happening always that we have this bump in or this maximum for intermediate migration rates. We wanted to figure out, when does this happen? Can we find any kind of simple conditions for under which, when do we expect migration or gene flow to be positive and when not? And so we started with a very simple model, like the simplest we can come up with. So it's just a 2D model where we have migration between these two populations. So team is just a subpopulation. And then we have some temporal change. So after a certain amount of time, we set the time to 0 at this point, one of these two environments is deteriorated. So it's we apply some environmental stress. And then after feeder generations, then we go into phase 2, then both teams are deteriorated. So this should mimic this diffusion of antibiotics through through the environment. And then we assume that so in the original habitat, our population is well adapted, and we need to adapt to this deteriorated environment. So we just say that a rescue mutation is adapted to the deteriorated environment. So it has a fitness 1 plus set, where set is a number larger than 0. So this is the absolute fitness. So it has a positive growth rate in this environment, and it has a negative this is the relative fitness, but it has a selective disadvantage to the wild type population. So essentially carrying the rescue mutation in the absence of any environmental stress means you're outcompeted by the wild type individuals. And then we have migration between these. This can be symmetric or asymmetric. I'll mostly talk about symmetric migration. So, and if we now look at a cartoon of how this model would look like, so now we have these two populations, team 1 and team 2. We have time on the y-axis, on the x-axis, on the y, we have the population size. And the colors show the different environments. So green is the original, red is the deteriorated. And the blue one is the wild type population size, and the black is the total population size. And we see that the wild type population goes down, gets extinct, and the mutant increases and leads to positive growth rates again. And the interesting part is this phase in between. So either both are in the original or both are in the deteriorated. And in between, we have this phase where we have a declining population size in one beam and more or less constant population size in the other beam. It's slightly smaller than carrying capacity. That's because, well, migrants continuously move out and then die here. So this is just a sketch of how these dynamics look like. And now we try to model this. And so the way we approach this is by just looking at a general formula for how we can calculate this probability for rescue. So, and this probability for rescue here is conditional on a team. So let's say we look at each team separately. So what's the probability that rescue occurs in one team? And then we can combine this very easily to a total probability, but it's easier to write the equation like this. And this is an easy way to write this. Is just to take this part here in the square brackets, which is the probability for no successful rescue mutation in any given generation. So which would be the mutation rate times the population size at a given time t in a team gives us the number of mutants that enter the population. And if we multiply this with the probability, pi, that this mutation actually is successful and isn't lost again due to drift, then 1 minus this is roughly the probability for not having a successful rescue mutation. If we multiply this over all times from -infinity to +infinity, we get the probability that there's no rescue event, and the inverse of that, 1 minus this, is then the total rescue probability. And because it's often, so this would be in discrete time, but we can approximate, or we can move to continuous time by just replacing this product with an exponential and an integral here. So, and the key ingredients when we look at this formula are the evolution of the population sizes in the two teams and the fixation probability of a mutation that occurs at a given point in time in a given team. So these are the two things we need to somehow model. I'm making a few assumptions here already, and but we'll see that they are fine. So let's start with the population dynamics. So we make it our life really easy because we want to have the simplest possible model and then later see how we can make it more complicated. So we keep the population size constant at carrying capacity K. So we model this with deterministic dynamics in the original environment. So we don't explicitly have to model density dependent growth and population regulation. And then we have a parameter R that says the population size declines at a given rate R if you are in a deteriorated environment. And then we can just write down a relatively simple differential equation that should describe the dynamics in phase 1 and phase 2 with the corresponding initial conditions. So like in these different phases. So that's just a little bit of boring algebra, but we can solve this explicitly, and we can get some expression for how these populations, the population of the wild type individuals, should behave. The other part is more interesting. So when we look at the establishment probability of rescue mutations, so the fate of these rescue mutations once they appear. And when we look at this diagram of our model, we have the phase 1, where it's a homogeneous environment, both are the same. And we have phase 2, where we have a homogeneous environment. So both teams are deteriorated. So if we use, let's say, branching processes to model the establishment probabilities of mutations, well, in the original environment, the rescue mutation is outcompeted by the wild type. So in large populations, the rescue probability, or the probability that such a mutation actually established in the population, will be 0. After phase 1, so in phase 2, the mutation has an post-selective advantage Z. So the fixation or establishment probability is simply 2Z, based on Haldane's classic results for branching processes. So what I've ignored here now is that mutations can also, that's true if you're in this state all the time or in this state all the time, but we have these transitions from one state to the other here. And we just ignore this just to have a simple model. So we just model the spatial heterogeneity in environments, but not the temporal one. And this should be a good approximation if these phases are long enough. And the interesting or more challenging part is figuring out what's going on in this case. So when we have mutations that are adapted in one environment, or adapted in one team, but maladapted in the other. So we can also model this using branching processes, but now we need to have a two-type branching processes where the two types refer to the location of the mutant allele. So type 1 is a mutant allele in the deteriorated habitat, so where it has a positive growth rate, and type 2 is a mutant individual in the original habitat where it has a selective disadvantage. And what one typically does in these multi-type branching processes is to write the average number of offspring in some form of table, and we can summarize this here. So a type 1 individual, so a mutant in the team where it's adapted to, has on average 1 plus Z times 1 minus m½ offspring. So why is this? So it has, after reproduction, it has a fitness 1 plus Z, so this is the expected number of offspring, and 1 minus m½ stay in the same team. So we use m½ instead of m here because so that we don't need to rescale when we talk about the stepping stone model. So essentially with probability m½ you move to one of your neighboring teams. And 1 minus m½ is everybody who stays. And then it can also leave 1 plus Z offspring, and then m½ of those become type 2 individuals because they migrate to the other team. And the same for type 2 individuals, except that the fitness is now 1 minus S. And so this is the expected number of offspring of these type 2 individuals. So a mutant individual in the green habitat. If we assume that the parameters are chosen such that this is a slightly supercritical branching process, what does this mean? This essentially means that evolutionary forces are not too strong. But in mathematical terms, it means that the eigenvalues of this, or the leading eigenvalue of this matrix is 1 plus something small, essentially. And then what we do is we rescale all the parameters with respect to Z. So we model all M and S relative to Z, and then essentially do this Taylor expansion in Z. So assume that the strength of selection is small, but leave the ratio of all these variables constant. So essentially we assume that all of these variables are not too large. So Z, M, and S should not be too large. This is very vague, but when exactly this is a slightly supercritical branching process is not straightforward to figure out, but that's roughly the conditions. And then at these conditions, so essentially if migration selection is not too strong, then we can calculate these establishment probabilities for team 1 and establishment probability for team 2, and they take the following shape. Essentially, they are an average over Z and S, where we have weights that depend on the relative contribution of Z plus S is the difference in the strength of selection between the two teams. So how different are the two habitats in terms of selection, and lambda is just something that measures the total magnitude of migration and selection in a way. So we have this nice weighted average. This looks a bit like this. So this is the establishment probability on the left-hand side for various combinations of parameters for mutations that occur in the team to which they are adapted to. So this is a rescue mutation occurring in a deteriorated habitat. So, and we see that this is a decreasing function of the migration rate. And if we make the selective disadvantage smaller, then, well, the establishment probability is larger. But overall, it goes from 2Z towards 0 for larger migration, which makes sense because, well, if you move out of the migration can only move you out of the right habitat. But in the, it's more interesting in the other team. So we find this result that, or this is, we find that the establishment probability is an increasing and then decreasing function. So there's a local maximum for intermediate migration rates. And this also makes sense because if you're a mutation and you're in the wrong environment, some migration is good because it can bring you in the right environment, but too much migration is bad because, well, then you essentially experience always two different, and like experience a mixture of two different environments. And this is another like interesting fact. We can connect this to classical theory for establishment or for local adaptation. So if we calculate the condition when these two probabilities are 0, so essentially when local adaptation is not possible, then this condition is the same as the one that has been derived in the 70s for gene swamping. So essentially, if migration is too strong relative to the strength of selection, then we will find no rescue mutations that can establish. Okay, so we can use this and we can follow up on this because we see now that there is also, this is, it seems like this advantage, and therefore intermediate migration rates somehow becomes important during evolutionary rescue, right? Because this is when you think back about the figure in the introduction, we also saw that sometimes you have this maximum for intermediate migration rates. So we want to see what is the role or what's the relative importance of these two lines. Like we have something decreasing in the migration rate and something with an intermediate optimum. So what we can do is we can calculate the rate of local adaptation in this system. We can make an average over these two establishment probabilities, where C is the relative size of team 1. That's in our notation, the one where the mutation is beneficial. And then we can ask, so this means what's the overall rate at which locally beneficial mutation establish averaged over both teams. So we allow them to occur in both teams. And then if we take the derivative of this with respect to m and evaluate it at m equals 0, so we want to know when is migration helpful, we find that either if the population size in the team where the mutation is actually detrimental is larger than the other one, so we have this asymmetry here, or if the gene flow from 2 is the original habitat to 1, the deteriorated habitat, if that's larger than the gene flow in the other direction. Okay, so if I just translate this into words, this would mean essentially whenever we have some source sink dynamics, where the sink, so the smaller population, receives lots of immigrants from a larger population, and local adaptation is necessary in the smaller population. So this is exactly this scenario here. So gene flow is beneficial for establishment of locally adapted alleles if we have a scenario where we have a large influx of individuals into a small or a declining population. And I think this makes intuitively a lot of sense. And now we have all the ingredients to see how this really carries over to a rescue probability. So we go back to this equation. We now have some formula for the local population size evolution. We have establishment probability approximation, so we can put all of this in. We can actually solve this integral, and we get a relatively long formula that I don't want to bore you with, but we can solve this analytically and in closed form. And here is comparison to individual-based simulations for a variety of parameters. The axis here, so on the x-axis, we have the migration rate. So on each plot, we go from small migration rate to large migration rates. And here on the y-axis, we always have the rescue probability. And then if we move from panel to panel here essentially we increase the either R, which means population size decreases more quickly. So this is the parameter that tells us how quickly the population size goes down. Or theta. Theta is the length of this period. Okay, so this if theta is large, then this period is long. And if R is large, then this goes down very quickly. So here we measure whether environmental change is severe or if it's slowly across space. So these two things seem to be similar in our model. And here we increase this, and you will see in a moment why I scale it like this. And here I have S divided by Z. S is the selective disadvantage of the rescue mutation, and Z is the advantage. So this means how costly is the rescue mutation relative to its benefit. So we have the cost here. And we see that across all these parameter combinations, we have a relative good fit between theory and simulations. So this vertical line just indicates the maximum migration rate. And the only range where it does not really work is here. We have this smooth transition and not a very sharp transition as we predict by our model. And this is exactly because we ignore that there's a temporal change in the environment as well. So we look just at these three phases separately and glue them together. And this corresponds to mutations that occur right before, like right around here, just before the environment changes, or right before here we have a temporal change in the environment. Since we ignore this, we don't get this moving out. But since we're mostly interested in like the evolution or the properties for relatively weak migration rates, or where we see this initial increase, so we were happy with this approximation. So now we can go back to our original question and ask, when does gene flow or when does migration actually facilitate evolutionary rescue? So when do we see an increase here relative to something flat? This is because it's on log scale, it's hard to see, but this is actually a decrease, so like it's a monotonic decrease. So the easiest way to do this is just to take the rescue probability that we can write down in closed form, take the derivative with respect to m, and evaluate it at m equals 0. So what's the slope at m equals 0? Is it going upwards or is it going downwards? And then we find this really simple condition. So if the cost of the rescue mutation is smaller-ish, then the R is the severity of the environmental change. So how quickly are individuals dying? And theta is, it's the how quickly is the change happening across these two themes. So how quickly are like antibiotics diffusing over space, if you want. And this ratio matters. And I think, okay, first have a look at how well this works. I think it works reasonably well. It's not perfect because there are a lot of approximative steps, but the blue part is here where this inequality does not hold, or where we have S divided by Z larger than R over theta, and the bottom triangle is where this condition holds. And it's not, as I said, it's not perfect, but it gives us a very good intuitive idea of what's going on. And here it's where this equality is already where we can have an equal sign in here. And so let's think about what this means. So if we go back to this, it essentially means if the rescue mutation is not too costly, and if this period is long, or if the line here, the population number drops really quickly, then we see a positive effect of migration on evolutionary rescue. And this is exactly what we found before. Essentially, we need a long period of asymmetry. Like we need a long period of source sink dynamics. Because when we have this source sink dynamics, then the contribution of the establishment probability of mutations that originate in team 2 is more important than the contribution of the establishment probability of mutants that originate in team 1. So I think that's a neat result. And I think this is useful because this can help us to design experiments, right? Where we try, okay, if we can control these 4 parameters, then we can create extreme scenarios and see if this is really, this really leads to a separation between 2 regimes where we have an advantage of migration and not, or not. Okay. So then a little bit of, we can generalize this. So we can also, this result was, it's purely about de novo mutations. So there's no standing genetic variation. We can also include standing genetic variation, or we can introduce asymmetry in the migration rates or in the carrying capacities. But let's look first. So standing genetic variation is easy. We just need to say, okay, at the beginning of the environmental change, there's a certain proportion of mutations that are already present. So this is like the mutation load that built up before. If we call this F0, then we can calculate this condition again. It changes a little bit. It gets a little bit more complicated. The mutation rate enters and so on. But all it does is diluting the any potential advantage that gene flow can provide. So that makes sense, because if there's lots of standing genetic variation already present, well, then you will get rescue from those, and gene flow doesn't really help or hinder it in any way. So this is really, standing genetic variation just dilutes the importance of gene flow. And then if we have some asymmetry, all we need to do is plug some quantity f in our equation. And this is really just measuring the degree of asymmetry. So this is the migration rate from team 2 to team 1 times the carrying capacity in team 2, and then in the corresponding thing for team 1 in the bottom row. And that, of course, if you have more asymmetry in the model, in the sense that the team that goes deteriorate first is the smaller one, then this will further amplify the parameter or increase the parameter range where we see rescue. Okay, so that is not too surprising. So if I just wrap up this first part really quickly. So we have seen that in this very simple model that gene flow facilitates evolutionary rescue if either environmental change occurs slowly, but it is severe, and rescue mutations are not too costly. So in other words, if the system shows source sink dynamics with more individuals migrating into the deteriorated habitat than out of it. Both of these conditions, or especially the first one, create scenarios that are like this. And then the second one relates more to gene swamping, because if rescue mutations are too costly, well, then gene flow will do more harm than good. And I think that also, if I can quickly go back to the introduction where I introduced these biofilms, this is a little bit what biofilms also create. They also modify the environment in such a sense that environmental change occurs slowly across space because we have this diffusive dynamics of antibiotics. So this could also shed some light on why biofilms seem to be so beneficial for the establishment or the evolution of treatment resistance. And for the last or second part of this talk, I want to extend some of these results to the island and the stepping stone model. So the paper by Hildegard Uecker that I mentioned in the introduction was focusing on an island model where we have migration between all possible teams. And we now take this model as our kind of null model and see how results change when we go to a stepping stone model. And the reason for this is that the island model can be reduced to a 2D model, where essentially the proportion of habitat 1 and the proportion of habitat 2 are the 2D. So we can, if we just have 2 habitats, all that matters is the gene flow between the 2 habitats. And so we again have this gradual change. So gray now means that the environment is deteriorated. So if you have this gradual change in the environment over space, except we don't really have spatial structure. But in the stepping stone model, where we only have migration between neighboring teams, we have the same environmental model, but the key difference is that now we have this spatial structure and isolation by distance. This is was getting way too complicated for us to model. We tried, we failed, so we did simulations. So if we just look at the stepping stone versus the island model, here the gray points are simulations point for the stepping stone model. The black ones are for the island model. And now we have 4 teams. So for 2 teams, these 2 models are of course equivalent. And here we have 16 teams. And we see that there's, in most cases, there are some cases where it's not true, but in most cases, in most relevant cases, biologically speaking, all we see is that the shape of the curves stay the same. So again, probability of rescue versus the migration rate. But as the more we increase the number of teams, the more the stepping stone model is shifted or like kind of stretched. It's like the same figure, but for smaller migration rates in a way. So we need much larger migration rates to get the same effect. And I think this was actually the hypothesis that we started this project with. And what we try to do is, can we rescale the migration rate to calculate an effective migration rate that is calculates not what's the probability that an individual moves from one team to the other, but what's the probability that it moves from one habitat to the other. In the island model, that's relatively easy. The stepping stone model, well, we can try to calculate this, and with some crude approximations, when we calculate an effective migration rate, we see that for 4 teams, the fit is perfect. For 16 teams, it's not perfect, but much better than before, and works very well for small migration rates. So this is mainly simulation work, so it's not super convincing, but in most cases, we really see that what matters is not the gene flow between teams, but the gene flow between habitats. And this is, if this holds true in general, if one could show this, this would actually mean that everything we learn from a 2D model can be extrapolated with some modifications to a multi-team island model. That's relatively straightforward. And then if we can also reduce the stepping stone model to something that behaves like an island model, then essentially it means that our results from 2 teams should be valid for a relatively broad range of models. There's another interesting effect of this. So in the island model, the amount of fragmentation, so that essentially by fragmentation, I mean the number of teams. So I take a total number of individuals that I leave constant. Let's say I have 10,000 individuals in total, and then I distribute them over a given number of teams. So I just say this would be 16 teams would be a more fragmented population because we have more isolated teams in the stepping stone model, and 4 would be just a relatively unfragmented population. But so in each team would have 4 times the number of individuals than in the case of 16. So I can plot the probability of rescue as a function of the degree of fragmentation or the number of teams if you want. And then if you do this for the island model, I maybe should have shown the figure, but it's boring because the lines stay flat, which makes sense. It doesn't really make much of a difference. This was already shown in 2014, that it doesn't really matter how many teams you have in the island model. There's a small change from 2 to 4, but then it stays really constant. In the stepping stone model, we see that it has a large effect. So the yellow line is weak migration, the blue line is strong migration. And the 2 interesting cases are this blue line here, and this purple line here. So what we see, if we have very strong migration rates and a small number of teams, this means essentially that we get gene swamping, mutations cannot establish because I choose a very costly one here, just to show this point very clearly. And if we increase the number of teams, then because of the increasing fragmentation, gene swamping becomes less and less likely, because even if you have a high overall migration rate, you mostly stay in the same habitat because there's just so many teams. If you go to a neighboring team, it's very likely of the same type. So migration doesn't do much in terms of evolutionary rescue. If you have intermediate migration rates, however, so that means you're at the optimum in a way in the 4-team model. So you're right, you have the demographic rescue from the inflow of individuals, and you don't have much of gene swamping. And if you increase the number of teams, essentially this demographic rescue effect from the inflow of individuals in the deteriorated environment gets reduced, so the probability of rescue decreases. So this means this interplay between the genetic effects of the establishment of mutations and the demographic effects of the population dynamics have an intricate interaction in the stepping stone model that we don't see in the island model. And so this is a preprint, so I'm not involved in this. This is I really enjoyed reading this paper, so I met Victor last week, and he showed me this preprint of his. He's a PhD student at the ETH in Zurich, and they studied local adaptation on graphs. And it's really interesting because so this parameter R theta that they look at measures the habitat heterogeneity. So essentially, if this is -1, then it means that we have this very strong negative correlation between neighboring habitats. So this would be a stepping stone model with just alternating themes. And here, if this R theta is very large, then we have a very strong correlation between the habitats of neighboring themes. So that would be more like the scenario we study. But in a way, this R theta is very strongly connected to the amount, like to how likely is it that two neighboring populations are of the same type. So it's very strongly related to the amount of gene flow between different habitats. And then we see this very, when we look at many different, very complex spatial models, we see that this is really what drives. So beta S is the amount of local adaptation. So you can either adapt to the blue or the red habitats. And this is really almost in a perfect linear relationship between the amount of the habitat heterogeneity, which is not exactly the same as we show, but it's tightly connected. And this is, I found this very interesting to look at it from this different angle. It seems to confirm some of our ideas. And so this brings me to my summary slide. So we've shown with this model that gene flow can facilitate evolutionary rescue if environmental change is severe, but it has to occur slowly across space, which makes sense. If it happens too quickly, then gene flow, the environment is almost homogeneous and gene flow doesn't do anything. But also there's a genetic, so this is the demographic part about population sizes. Also rescue mutations have to be sufficiently beneficial relative to their cost. And for many antibiotic resistance mutation, research shows that the cost is relatively low. So this also is a condition that is probably very often true in these settings. And spatial structure, or the nature of spatial structure, matters because the stepping stone model, even though it shows qualitative similar behavior as the island model, gene swamping becomes less likely and gene flow hence becomes more beneficial by and large. So the effective migration rate between habitats is the key factor here. And the degree of fragmentation is also more important in a stepping stone model as compared to the island model. And I think that's a good point to stop. I also promised to talk about continuous space, and I have some slides prepared, but I think that's a good point to leave that maybe to the discussion if there are some questions.